Hi everyone, so today we're going to go through the thinking practice questions on Unit 1 that you guys completed yesterday. So let's start with number 1. If the following molecules were to undergo a dehydration synthesis reaction, what molecules would result? First of all, remember dehydration means the loss of water, okay? So you're thinking about a anabolic reaction where there is the loss of water as a result of the joining of two different molecules. Circle the part of each amino acid that will interact and draw the resulting molecule. So you have amino acid 1 and amino acid 2. You should know that the OH here will join with the H of the amino side. So the carboxyl group will lose an oxygen and a hydrogen, and the amino group will lose a hydrogen. So 2 hydrogens plus 1 oxygen will give out an H2O. So it says, what is the resulting molecule supposed to look like? Well, if you lost the oxygen and the hydrogen here and the hydrogen here, then you only have a carbon and a nitrogen. So both the carbon and the nitrogen will bond, creating this peptide. Okay, you didn't have to mention that these were R groups, but if you knew that, that was awesome. Okay, it says, identify which of the 6 main elements, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, are found in each of the 4 macromolecules. So you should know the macromolecules are the carbohydrates, the lipids, the proteins, and the nucleic acids. So in carbohydrates, as the name suggests, carbohydrates, there should be a carbon, a hydrogen, and you should know that there's also an oxygen, and it's usually in a 1:1 ratio. So glucose, for example, is C6H12O6. So for every 6 carbons, there's 12 hydrogens. So there's a 1:1 ratio. Lipids contain almost only carbon and hydrogen, very, very little oxygen. That's why they're hydrophobic. Remember that if you contain very little oxygen or very little nitrogen, which are very electronegative, then you are hydrophobic because you do not have a charge. Okay? Proteins contain carbon, hydrogen, oxygen, nitrogen because of the amino group, we just saw it here, and it can also contain sulfur, okay? And it's not going to contain sulfur in the main structure, but it can contain sulfur in the R groups, hence disulfide bridges that you've probably already reviewed in protein structure. Then nucleic acids, carbon, hydrogen, oxygen, nitrogen, and phosphorus. Remember that phosphate backbone that nucleic acids like DNA and RNA have. Okay, question number 3. Describe the relationship between substrate concentration and reaction rate shown in the graph below, well, in the right, and propose an explanation for it. So you're seeing substrate concentration, okay? Substrate concentration can increase, but reaction rate is going to increase at one point and then stabilize, okay, or plateau. This is called a plateau. As substrate concentration increases, the rate of reaction increases because there are more substrate molecules for the enzyme to act upon. However, once all enzymes are saturated, so at this point, let's say that all enzymes have been saturated with substrate molecules, they're all doing the work, there's no more enzymes to increase the reaction rate, the rate of reaction cannot increase any further. So it plateaus right over here after enzyme saturation. Okay, DNA polymerase from T aquaticus. Oh my god, I'm so sorry. DNA polymerase from T aquaticus. And this is actually something that I know a lot of. It's TAC. It's used in PCRs, okay? Is used in PCR, so it's a DNA polymerase from a particular bacteria. PCR is a technique where millions of copies of DNA can be made from one original copy. In this method, the target DNA molecule is subjected to temperatures over 95 degrees Celsius to make the double-stranded DNA separate. So in the moment that you heat it up, the reason why you heat it up is so that you can make the double-stranded DNA separate, and then you can include the DNA polymerase to start polymerizing that DNA. The temperature is often then lowered slightly to allow primers to anneal or to join or to bond let's say, before the Taq polymerase catalyzes the reaction to incorporate new nucleotides into the complementary strands. The cycle is then repeated over and over until there are millions of copies of the target DNA. Predict why this bacterial polymerase is used instead of a human polymerase. So again, go back to the data. What is it telling you? It's telling you that this method of PCR, the target DNA molecule is subjected to temperatures over 95 degrees Celsius. Okay, the Taq of this DNA polymerase, the DNA polymerase of this Taq organism actually comes from, this organism is heat resistant. So it can work and it can replicate DNA even when there's a lot of heat. Our human body can't do that. When we have a fever, we start, it's very mortal for us and very dangerous for us because no enzyme in our body actually can work past 40 degrees Celsius. So it can withstand high temperatures without denaturing. So we use this Taq DNA polymerase instead of human polymerase because it can withstand high temperatures. What would happen if you used a human polymerase in a series of PCR reactions? The DNA strand will separate because it's going to separate because of the 95 degrees Celsius, and the primers could anneal, they could bond because you're decreasing the temperature at the time that the primers bond, but the polymerase is not going to be able to copy the sequences because the minute that the temperature rose to 95, our DNA polymerase would have become denatured. Okay. Imagine a protein chain that includes the following amino acids among several others. So there's serine, cysteine, asparagine, and phenylalanine. Okay? Which of the amino acids could form a hydrogen bond with another amino acid in the chain to stabilize the secondary structure of B-pleated sheets? So at this point, this is a very technical question, but you should have already seen what affects the the structure, the secondary structure. So I'm going to take you right now to this image. Okay, so what you can see is that in the secondary structure, you can have B-pleated sheets or you can have alpha helix sheets. So you don't want to see this helix sheets, you want to see this B-pleated sheets. And basically what it's showing you is that to be able to form these sheets, the hydrogen of an amino group, okay, of one of the amino acids needs to bond to the oxygen of a carboxyl group of a non-adjacent amino acid. They're not amino acids that are next to each other. They're amino acids that are kind of one on top of another. So this hydrogen of the amino group will bond to this oxygen of the carboxyl group. As long as any of your amino acids have an amino group and have a carboxyl group, they're going to be able to form hydrogen bonds to be able to create B-pleated sheets in the secondary structure. So let's go back to the question. Which amino acids could form a hydrogen bond in the chain to stabilize the secondary structure of the B-pleated sheet? The answer is any of them. Okay, any of these amino acids, because all of them have an amino group and a carboxyl group, hence all of them could potentially create hydrogen bonds for B-pleated sheets in the secondary structure. Which of the amino acids could form disulfide bonds with another amino acid in the chain to stabilize the tertiary structure of the protein? This should have been a little bit easier. Disulfide, sulfide. If it's telling you it's sulfide, it's telling you it has to contain sulfur. So you would have to see which of these amino acids would contain sulfur in their R groups. This one does not. It's just carbon, hydrogen, and oxygen. This one contains sulfur. Okay, so cysteine contains sulfur. This one does not, and this one does not either. Whenever you're seeing a ring-like structure like this, that means that these bends over here, these corners are carbons and they contain hydrogens attached to them. So this is just a carbon and a hydrogen. So the correct answer would be cysteine amino acids. Two cysteine amino acids together could form a disulfide bond. Question C says, which of the amino acids could participate in hydrophobic interactions with another amino acid in the chain to stabilize the tertiary structure of the protein? So hydrophobic, remember hydrophobic would mean that that amino acid has no charge. Okay, if you, or no slight charge, partial charge, okay? So what you're looking for is something that's kind of lipid. So if you're thinking about lipids contain carbon and hydrogen. They do not contain a lot of oxygen. They do not contain a lot of nitrogen or other molecules, because those elements are very electronegative, so they're going to have a strong pull on the electron of different elements, and they're going to create slight negativity, okay? So right over here, you can see, for example, that this contains an oxygen, okay? So this is probably going to give you a charge. Here, this contains a sulfur, right? Here, this contains an oxygen as well. And here, you can see that this is carbon and hydrogen. And as I told you guys before, a ring-like structure signifies that there's only carbon and hydrogen here. So the correct answer would be cysteine. Okay, cysteine, because it doesn't contain oxygen and nitrogen. It does contain sulfur, but sulfur does not have that much of an electronegativity. And phenylalanine. Okay, so both cysteine and phenylalanine will have nonpolar R groups, so hydrophobic interactions can form between these amino acids. Okay, and the last question, short response question. Water is important for all living organisms. The functions of water are directly related to its physical properties. Describe how the properties of water contribute to two of the following. So you have three, so you only need to talk about two. Transpiration, remember, it's the way that water moves through a plant. Thermoregulation and endotherms, so the regulation of organisms that regulate their body temperature internally, not with the external environment, but internally through metabolic reactions. Plasma membrane structure. Okay, so 2 points for each process, 1 point per category in the context of linking properties to contribution. So okay, so you want to have 4 points maximum. Okay, so transpiration. What could the property of water be to help transpiration? Polarity and cohesiveness. Remember, cohesion allows you to a body of water, to move cohesively. Light heat, a high heat of evaporation, that has to do with evaporation in plants. Water potential, okay, that has to do with the movement of water through roots into plants. Contribution to the process, cohesiveness allows water to move. High heat of evaporation reduces water loss. Water potential allows also water to move from roots to the plant. Okay. Thermoregulation. So again, high heat vaporization, that's actually how a lot of organisms can keep cool. So remember, evaporative cooling, you can get rid of a lot of the highest or the warmest, the most hot, and in that case, excited water molecules in the surface of your skin. And if you get rid of the most excited water molecules in the surface of your skin, you're going to decrease, this is all in Unit 1, remember, you're going to decrease the kinetic energy of the water molecules that are staying in the surface of your skin, and that allows you to cool down your body and cool down your skin. So if you do not 100% recall how the high heat of vaporization leads to evaporative cooling, all of the information is there in Unit 1. High specific heat, you also takes a lot of heat to warm up water, so that allows you to buffer or kind of balance out and control the amount of heat that is in your body, since a lot of your body is made out of water. Plasma membrane, the polarity, okay, so the arrangement of phospholipids is also super important when it comes to controlling the water outside and the water inside, or the liquid outside and the liquid inside of the plasma membrane. Okay, so hopefully that makes sense. If it doesn't make sense, if you want any extra help in any of these questions, please send me an email, and thank you very much.